Great. For everybody online, thank you so much for joining us for today's HMSC research seminar. My name is Cinnamon Moffett, and I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center located in Newport, Oregon, and I will be your host for today. If you haven't been with us before, just a little reminder that your cameras, mics, and screen shares have been disabled. But for today's speaker, we'd really like you to put in any questions you might have into the chat whenever they come to you, and then we'll answer them at the end. But feel free to interact with us that way by using the chat function. By now, everybody knows where it's at, most likely on Zoom, but if you're not familiar with Zoom, you'll either find it at the bottom or the top of your screen. And you can just click on the pop-up box. I also wanted to let folks know that we are recording today's event, and you're able to share that or listen to it again if you're missing something. And I just put the link to where that's going to be into the chat. I have a couple of quick announcements, and then we'll get started with today. For those of you that are online and that are familiar with Hatfield Marine Science Center, I just want to let everybody know that we're going to take a short break from our seminar schedule, but we'll start again in the fall after the fall term begins. There's a few things still in flux, but our hope is when we come back, we'll be able to do a seminar in a hybrid format. And so that means when the speakers can be on site, the audience can also be on site or remote. And if the speakers are fully remote, then we'll be fully virtual. This will take a bit of communication and coordination, so please watch the details of the seminar announcements. All of the details on what we're doing and what format we're using will come out there. The other thing I wanted to let folks know is that if we are on site, seminars will hopefully take place in the new Marine Studies Building Auditorium with COVID restrictions in place. And so what that means is that if we are on-site option, on-site will only be available to folks from HMSC and our co-located partners on our campus. Anybody outside of HMSC will have to join us online. That is a COVID restriction from OSU and that we're trying to meet. We'll also be reducing the capacity in the auditorium to provide a 6-foot distance, so there'll be plenty of room for folks at Hatfield, but it might change where you might sit in our new auditorium. And so there's not a mutiny at the door. I'm warning you all now, we will not be serving coffee or cookies for at least a little while at the beginning of seminar because face covers will be required while we're inside in the auditorium. So again, COVID restrictions and the functionality of the Marine Studies building aren't quite confirmed yet, but my fingers are crossed, and I'll let everybody know more details as announcements come out. If you are interested in figuring out what's coming, you can go to our homepage, HMSC, scroll to the bottom, and there's a calendar of events there. That is where you'll find any of the information about whether it's going to be a remote or a hybrid event as we move into the fall. I also wanted to let folks know if you're missing learning opportunities during this little seminar break, we have several research talks from our ecosystem oceanographers and our ocean ecology candidates for the 2 new positions that will be housed at HMSC. And in fact, we have 2 next week. One will be Tuesday at 11 a.m. and the next one will be Thursday at 9 a.m. So please make sure to check them out and watch for the announcement from the other candidate talks in the next few weeks. But you don't have to go without learning opportunities while we're not doing seminars. So just kind of keep an eye on that. All right, for today, let me tell you a little bit more about our speaker. Megan Considine will be giving a seminar about shell boring polychaetes in the Pacific Northwest oyster aquaculture industry. Megan graduated from the University of South Carolina with a bachelor's degree in marine science, during which time she found her passion for oyster restoration work and outdoor education. She then pursued her master's degree in marine resource management at Oregon State University and graduated and finished her defense last month. So congratulations, Megan. During that time, she worked with the Oregon oyster aquaculture industry to detect and prevent emergent nuisance species that harms product quality. Megan currently is working with coordinating the Olympia Oyster Enhancement Project in Yaquina Bay, right around the corner from us here at Hatfield. So we're excited to have Megan here, and she's got a lot going on. She's finishing up the last little pieces before she starts a new job, which she just officially started today. So thank you so much for taking time to join us. Megan, the floor is yours. I hand it off to you.
Thank you so much for the introduction, Cinnamon. So today I'll be telling you all about the work I've been doing the past 2 years for my thesis, which has focused on shell-boring polychaetes in the Oregon oyster aquaculture industry, which has been a part of a larger project focused in the Pacific Northwest. And before getting started, I wanted to first acknowledge the 35 partners on this multi-state project. These folks provided help and support that made this project possible. So I'll start with introducing the Pacific Northwest oyster aquaculture industry, and then I'll give you some background into shell-boring polychaetes, specifically how to identify them, how they devalue oyster products, and the likely vector for their introduction. And then I'll get into the larger project goals, which includes mapping the distribution of mudworms, determining variables influencing prevalence of infestation, identifying effective treatments, and as well as research implications for various stakeholder groups. So shellfish have been harvested in the Pacific Northwest for thousands of years. And oyster aquaculture has been important economically and culturally since the late 1800s. Today, oyster aquaculture in the Pacific Northwest is a multi-million-dollar industry, generating $270 million annually, making this region the leading producer in bivalve shellfish within the United States. This industry provides jobs to over 3,000 people and is culturally important to local communities, Native American tribes, recreational harvesters, and family-owned farms that are involved. The oyster of focus for aquaculture purposes in the Pacific Northwest is the non-native species Chrysostrea gigas, or commonly known as Pacific oysters. There are two main markets for oysters, which are canned meat and oysters on the half shell, which you can see presented in the two photos on the right. Oysters on the half shell are much more lucrative than canned, and there's also a growing consumer demand for raw oysters served in this fashion. The majority of growers in the Pacific Northwest do sell to the half shell market, and this region is prized for its oysters that often have a consistent shell color. Recently, researchers and oyster farms in Washington state started noticing that oysters were starting to become infested with a pest that changes the appearance of their inner shell. So these pearly white shells, often have bits of mud or darker purple coloration, which are common. The change in appearance begins with the initial infestation when the pest burrows into the shell. And these burrows over time can become blisters, which are bumps inside the shell that fill with anoxic mud and fecal material. And sometimes these blemishes can become punctured when shucking the oyster, which affects presentation and taste. The small pest responsible for these unsightly blemishes is a parasitic polychaete, Polydora webstri, as well as other Polydora species, and they're more commonly known as mud blister worms. The worm and the mud inside the blisters is not harmful to human health, but as you may have already guessed, these unsightly blisters can render the lucrative half-shell product unmarketable, and it's not only an aesthetics issue. Spencer and colleagues did a recent synthesis and summarized the impacts of shell-boring polychaetes on host fitness, which can include decreased growth, decreased shell strength, meat weight, as well as impacts to other physiological characteristics, and even in some cases, increased mortality events. And these challenges for oyster industries have been seen worldwide. Polydora infestations have caused substantial economic losses and have even been responsible for the collapse of aquaculture industries in different locations all over the globe. Recent research by Rice and colleagues in 2018 found that populations of mud blister worms in the Pacific are genetically similar to populations in the Atlantic. So this suggests that the spread of the pest is probably due to other human-mediated processes and not just natural reproduction and dispersal. Which leads me to what has been suggested to be the most likely pathway contributing to the introduction of shell-borne polychaetes, which is the transfer of shellfish between countries, states, and even between estuaries within a state. And oyster transfers aren't only a vector for shell-borne polychaetes. Oyster aquaculture has resulted in 18 oyster species being introduced to 78 different countries. And these introductions can be a vector for shellfish diseases and other non-native species that hitchhike a ride into estuaries. Oyster aquaculture has been identified as the second largest vector behind ballast water globally for the introduction of non-native aquatic species. And in the Pacific Northwest, over 71% of the aquatic invasive species have entered through oyster transfers. So there are different regulations that can assist with controlling and preventing introductions, which is something that I'll get into a little bit later in this talk. So the researchers and producers in Washington that initially discovered these infestations on commercial oyster farms did confirm through molecular analysis that Polydora websteri is present in Washington state. And this research sparked the start of a larger multi-state project. So it leads me to some project goals I wanted to share. So to better inform mitigation strategies, researchers in Washington wanted to determine the current distribution and prevalence of shell-borne polychaetes in the larger Pacific Northwest. So that includes California, Oregon, Washington, and Alaska. And this was done by working directly with producers and harvesters on oyster farms. We also wanted to better understand what variables may be influencing prevalence of infestation. And once we know the distribution, it's important to also identify treatments growers can use to reduce transmission and mitigate impacts. And another goal was to identify research implications for various stakeholder groups. So I'm going to share with you results from these project goals related to the multi-state effort. And my part within this larger project has been focused specifically in Oregon. So starting with the first goal, in order to create a map of the distribution of Polydora species, we need to start by collecting oysters at farms. So my advisor, Steve Rumrell, and I collected 25 to 50 oysters per farm during each sampling period. So summer 2019, winter 2020, summer 2020, and winter 2021. We looked at oysters from different growth methods, so whether they were grown on the bottom or suspended off the bottom. And then I processed the oysters and recorded shell measurements, so that included length, width, depth, and thickness, as well as tissue weight. And then extracted any worms that I could find to send to Washington for DNA sequencing and species ID. And that processing was done from a variety of locations, this photo being my kitchen table. And any oyster that had at least one burrow or blister on either the right or left valve was considered to be infested. And then prevalence was calculated as the proportion of infested individuals for each sample. So again, my part within this larger project has been focused in Oregon. So I'm going to show you a map from the winter 2020 and summer 2020 seasons for the Oregon data. The bluish green and orange color are denoting the on and off bottom culture. Each arrow is a sample of 20 to 25 oysters and shows the prevalence or percent of infested oysters for that particular sample. So for the past 2 winter and summer seasons, we've collected both on and off bottom cultured oysters from 7 farms spanning Tillamook, Netarts, Yaquina, and Coos Bay. This season, we're seeing some signs of infestation in Tillamook Bay with that on-bottom sample at 8%, 8% prevalence of infestation by mudworms, and Coos Bay with one of the on-bottom samples at a 5% prevalence. Yaquina Bay for this season, both samples were at 0%. And then for nitards, we're seeing a high prevalence for both the on-bottom and off-bottom. And this winter 2020 actually had the highest prevalence that we saw for nitards during the whole project period. So now we're seeing the summer 2020 results. This time the prevalence was at 0% for both Tillamook and Coos Bay. But now we're seeing some signs of infestation for Yaquina with that on-bottom sample at 8% and off-bottom at 4%. So across seasons, there have been some signs of infestation for each estuary. And as you may have already noticed, nitards again has a higher prevalence, which was a consistent observation throughout the study. So this season, we're seeing 84 and 100% for the on-bottom culture, and then 40% for the off-bottom. And another observation, particularly for this season, is that it looks like there may be a difference between on and off-bottom culture, which is something I'll get into shortly. I wanted to also mention that in summer 2020, we discovered signs of infestation in native clam species in Nitards Bay and Tillamook Bay. Specifically, we looked at little neck clams from Nitards and butter clams from Tillamook. Overall, the sample size was small and I wasn't able to find and extract worms for species ID, so it's definitely something to look more into. If native stock is also infested, that has further implications for resource management. So why are we seeing such a high prevalence in nitards? It could be related to the history of oyster translocations in the area. Oyster aquaculture in Oregon started in the late 1800s with the importation of non-native Pacific oysters. And it's possible that growers in this estuary imported oysters from an infested area. Unfortunately, we don't have this information, so we aren't able to confirm this. It could also be related to differences in estuarine environmental conditions. For example, past studies on mud blister worm infestation have shown that prevalence is higher with lower salinity. Interestingly, on average, nitards has less of a freshwater influence than the other estuaries sampled, which is kind of the opposite of what one might expect given the high infestation. Studies have also shown that mudworm larval development and growth are temperature dependent, with slower development in cooler climates, and some species can stay in the water column for up to 85 days prior to settlement. Which brings me to residence time, because larval retention within an estuary is largely influenced by how quickly the estuary flushes. Interestingly, nitards has the shortest residence time of all the estuaries sampled. So that means there's less time for mudworm larvae to settle before being taken out to sea. So I did not record any environmental conditions during this study, so I can't definitively say what is really influencing infestation at nitards. But when looking at existing environmental data for nitards and reviewing existing literature on what environmental factors influence infestation, the high prevalence at nitards is kind of a head scratcher or somewhat non-intuitive. One environmental condition that more so fits the puzzle could be siltation levels. Literature suggests that higher siltation levels correlates with a higher prevalence of mud blister worms. And like many Oregon estuaries, nitards has experienced high levels of siltation due to the logging industry. Future research should collect in situ water quality measurements and perform spatial analyses to determine the influence of environmental conditions on mudworm infestation within and across estuaries. So the next project goal was to determine variables that may influence prevalence of infestation. So to tackle this goal for the Oregon data, we started by using a permutational analysis of variance with the factors of season, estuary, farm, and culture type, and the covariates of shell length and tissue mass in order to test for significant differences in mudworm infestation among those factors and covariates. So we saw earlier that there were some major differences for the different estuaries within Oregon. Other significant terms and interactions from this model were subsequently investigated using pairwise comparisons to determine which pairs differed significantly for infestation by mudworms. And so we'll start with the factor season. So the figure you're seeing here is essentially a hybrid of a box plot and a kernel density plot. So the width of the shaded area represents the proportion of the data located there. And so what you're seeing here represents the difference in prevalence of infestation for the variable season. Mainly what I want you to notice is the black diamond, which represents the mean. P-values in the right-hand corner were obtained from pairwise comparisons and are telling you if there are significant differences in prevalence between summer and winter. And what we're seeing here is that there's no seasonal differences in prevalence of infestation. So future studies may need to have a longer study period to really pick up on these differences. So now you're seeing the difference in prevalence of infestation for the variable culture type. And what we're seeing here is that there is a difference in culture type, with the off-bottom culture being 30% lower in prevalence than on-bottom culture. And we're thinking this may be due to differences in siltation for these culture types. As I said earlier, past studies have shown that higher siltation correlates with higher prevalence. And this is really an interesting finding because it points us to a potential mitigation strategy for growers to implement on their farms. I also looked at differences in oyster metrics of tissue mass and shell length. So shell length, for this figure, shell length is on the y-axis, x-axis is infested and non-infested individuals. And what we're seeing here is that individuals that are infested are 19% smaller in length. And so now tissue mass is on the y-axis and the x-axis is still infested and non-infested individuals. And again, what we're seeing here is that individuals that are infested are 46% smaller in tissue mass or meat weight. And so this is kind of a chicken or the egg scenario. It could be that smaller individuals are more susceptible to infestation, or it could be that worms are causing decreased growth. And so with the observational data collected, I can't definitively say the causality of the relationship. To try and understand this relationship further, though, I also looked at differences in tissue mass and length for the different stages of infestation. So whether it's a blister, burrow, or nothing. And we're seeing here that oysters with blisters, that later stage of infestation, are significantly lower in length than oysters with burrows, an earlier stage, or than for oysters with no infestation. And it's the same story for tissue mass. Oysters at a later stage of infestation show reduced tissue mass. And so while I can't definitively say causality based on existing literature and the stage of infestation information, we're thinking that infestation causes decreases in oyster growth because there's a shift in energetic demand. So as infestation gets worse or gets to that later stage, the oysters are put more energy towards growing shell around the pest rather than growing in tissue mass or length. And this line of thought takes us back to a previous slide about product devaluation. If oysters are smaller and weigh less, growers are going to get less money for their product. And so for the growers that have an infestation on their farm, the next big question is, what can I do about this? So in response to these questions, project collaborators in Washington decided to test various treatment methods targeted specifically for Pacific oysters in the Pacific Northwest. So the first treatment method was drying oysters for 2 days in shellfish bags. The second treatment method was placing oysters in a fridge for 3 days. And the third treatment method was a freshwater dip for 1 hour, followed by drying oysters in shellfish bags for 2 days. And the results for each of these treatments are shown on this graph. So the y-axis is the number of live worms per oyster after each of the associated treatments were applied. And the x-axis is the control, so no treatment applied, as well as the 3 treatments that I told you about previously. And you can see here, and you can see the researchers did these experiments both in the fall and in the spring. And so for each of these treatments, there was a 100% survival of all oysters, and the majority of oysters that were infested had dead worms after each treatment. So essentially, each of these treatments are viable options for growers and managers. And given that each of these treatments are relatively effective, really any one of them could be applied on a farm or could be used for management purposes in terms of oyster transfers. The route that each grower takes will largely depend on the resources they have available. So for example, the fridge method will require growers to transport oysters to a facility with electricity, while the fresh dry method requires access to fresh water and the ability to frequently change that fresh water as it'll get saltier with each batch. And the dry method may be one of the easier options because some farms may already have oysters airly exposed during low tide. So it's just naturally having that treatment applied. And so that brings me to the last project goal. In a 1998 paper, Lubchenko discussed how science should advance and return benefit to society and how scientific knowledge is urgently needed to make informed policy and management decisions. And with this perspective, the project team wanted to identify the implications of this research for policy and management decisions, as well as industry best practices, and provide potential options for these associated actions. So for the Oregon portion of this project, we are coming at this goal in several different ways, including developing a policy brief, as well as several outreach products geared for different audiences. So again, my part within this larger project has been focused specifically in Oregon. So I'll be sharing with you several Oregon shellfish regulations that could be utilized or revised to mitigate further spread of the worm. And this policy review could also be applicable for other states in the Pacific Northwest as well. So the major authorities for these regulations are the Oregon Department of Agriculture and Oregon Department of Fish and Wildlife. The Oregon Department of Agriculture's regulation that is applicable to this research is the leasing of plots for farming. And the state can place special regulations on what happens on those plots. So that could be requiring growers to avoid sensitive habitat like eelgrass, or it could be the state specifying which culture methods can be used, so whether that's on or off bottom culture. And from our research and other mudworm studies, off bottom culture has been identified as a potential mitigation strategy. But within Oregon, approval for off bottom culture can be difficult due to the associated trade-offs, which includes the shading effects that off bottom culture has on native eelgrass, as well as this not in my backyard phenomenon where waterfront owners don't want their views obstructed. So off bottom culture can be more visible and look more kind of unnatural than on bottom culture. And this not in my backyard phenomenon has limited oyster aquaculture growth in other parts of the US as well. And so a way to potentially mitigate this sort of attitude could be increased transparency with waterfront owners about the issues and benefits associated with these culture types. The Oregon Department of Fish and Wildlife deals with permitting for interstate oyster importation, so oysters coming from out of the state. The permitting process requires inspection for diseases and pests by an authorized shellfish pathologist, such as Aquatechnics in Washington. Currently, only interstate oyster transfers are required to receive a permit, and there is not a requirement for intrastate transfers. So, for example, growers in Netarts Bay, where prevalence is high, are able to move oysters to other estuaries within the state of Oregon without regulatory inspection and approval. So a change to this policy to include intrastate transfer could prevent not only the spread of shell-bearing polychaetes, but also other non-native species and diseases, such as the Oyster's herpes virus, which is currently present in Southern California. ODFW also regulates the importation of oysters from shellfish restricted areas. The designation of these restricted areas are largely for sanitary or water quality purposes, but this regulation could be utilized for limiting the spread of invasive species and disease as well. For example, if Netarts Bay was deemed a restricted area due to the high prevalence of shell-borne polychaetes, the state could require oysters originating from Netarts to have effective treatments done prior to transportation. So my advisor, Steve Rumrell, has been working with ODFW Shellfish Program staff and the ODFW Commission to revise some of these policies. And while these revisions may add more red tape for growers, many growers actually already implement them because they want to continue business with Washington State, where policies such as this are already enforced. So the statutory work that needs to be done in order to revise regulations that could address the problem can take a lot of time, and potentially time we don't have. So extending beyond regulatory agencies and reaching different segments of the industry, such as growers and processors, is a viable option for mitigation. So informing them of ways to identify infestation would be a great first step. Some of the growers that I worked with on this project were not aware their farm was infested prior, and now that they are aware, they're able to make or able to take those necessary actions. Informing growers of the pest's current distribution is another important piece of information. If growers know that Netarts is infested, then they know that they'll have to take extra precautions when transporting oysters from that area. Talking with growers about potential risks, such as decreased growth, aesthetic problems, and the ways in which the worm spreads, are all also important pieces of information that can help growers to safeguard their product. And of course, treatment options are also very important, especially for the growers that already have an infestation on their farm. So we're currently drafting a brochure to help disseminate this information, and we're hoping that this brochure will reach growers and processors that are not already engaged on this project. Beyond working with growers, focusing on consumer demand was an aspect that hasn't really been explored. In general, there's a lack of research on oyster aquaculture supply chains. And given that this mud blister worm issue is largely an aesthetically driven marketing issue, there's potential to tackle this problem through engaging with retailers and food services. A recent study by Love and colleagues identified personal relationships and trust between growers and retailers as important business values. The study also found mismatches in product attributes between retailers and growers, such as aesthetics. So for example, chefs wanted their oysters to be perfect all the way through, whereas growers recognize their product will have imperfections. There'll be barnacles and worms. And the growers in the study who sold through direct supply chains, like the top panel in this figure, and worked directly with chefs, were actually able to manage those expectations. An example of this consumer-focused solution can be seen with one of the growers on this multi-state project. This grower was hit particularly hard by mud blister worm infestation on their farm and ended up having to shut down their business. Later, the same grower was able to open back up by partnering with a local chef who specializes in making tasty cuisines from trash fish or bycatch, such as this wolf eel that was accidentally caught in a crab pot. So they created a chef pop-up at the farm, which resulted in lines out the door from visitors who traveled from many different locations. And so following this as a potential solution, graduate students at Portland State University created a consumer-focused outreach product that you can see here on the right to inform consumers that the worm and the blisters are not harmful. The thought is that this product could be used as a card for growers to have on hand and give out if infestation was particularly bad on their farm. And since there largely isn't currently an outcry from consumers in the Pacific Northwest, the project team decided that consumer-focused outreach could be dispersed and discussed internally with project collaborators until circumstances warrant. So to summarize the major findings for this project that I went over, for the first project goal relating specifically to Oregon, we found a 2.4% prevalence of infestation by mudworms for Tillamook Bay, 71.9% for netarts, 2.88 for Yaquina, and 1.11 for cues. Moving forward, future research could determine the influence of environmental conditions on mudworm infestation and also determine prevalence of infestation in native bivalves. For the second project goal, we found that off-bottom culture was significantly lower in prevalence of infestation compared to on-bottom culture. Infested oysters were also significantly lower in tissue mass and length. And when we teased that apart further, we found that oysters at a later stage of infestation were significantly smaller when compared to oysters at an earlier stage or with no infection or infestation. Based on the literature, we think that there's likely a change in energetic demand for infested oysters, but future research could investigate the mechanisms behind these observations further. For the third project goal, PIs in Washington identified 3 effective treatment options that could be utilized by both growers and resource managers. One of the next steps for this goal is to see how treatments really scale up on an actual farm. And for the last project goal, we identified several Oregon state regulations that could assist in limiting the spread of shell-boring polychaetes. And policy revisions could include intrastate transport permits and the implementation of shellfish restricted areas for pests and disease. And we also identified outreach and engagement with key stakeholder groups such as growers and consumers to be a strategy to mitigate spread and economic impacts. Next steps include synthesizing this research into outreach products to be distributed, and future research could include improving our understanding of how this issue propagates through the oyster aquaculture supply chain. And so with that, I'll leave it open for questions. And if you would like to talk further, here's my email.
Thank you so much. We definitely have a couple questions coming in, and I have a whole bunch myself if I get the time. So for everybody online, I'm actually, if you want to raise your hand, we're a small enough group that I can unmute you and can ask your question verbally, but we're also getting some questions in the chat, so we'll start there. So Megan, how is residence time in Netarts Bay determined? Some areas where oyster releases occur almost never empty of water from the tide flats because of the geomorphology and other factors. So can you speak to that a little bit?
Yeah, that's a great question. So I determined residence time just from like reviewing past literature, but I didn't look at it spatially throughout the estuary. And we also don't have exact coordinates of where the farms are located. So that's definitely a really interesting aspect to look more into, and a spatial analysis would better answer that question for sure.
So Megan, I had a question. I know that your project was really Oregon focused, but what was the patterns and trends that you were seeing on both sides of us?
Yeah, so in California, Washington, and Alaska, we're seeing signs of infestation in all of the estuaries that were sampled. Interestingly, Oregon's very unique in that Netarts Bay had the highest prevalence of anywhere sampled throughout the project. And then the other estuaries in Oregon were much lower compared to other estuaries in like California and Washington. So it's much more variable in California and Washington, whereas here it's like Netarts is definitely a hotspot.
Okay, I have another question for you. So I'm going to be selfish, everybody, as you're working on your questions. So for the treatments, and I know, again, this wasn't your project, but I was wondering about susceptibility of reinfestation after a shell has already been infested once. Has there been any work to understand that level of susceptibility if an oyster has been infected and then treated and some of the worms removed? Is there a chance that they'll be reinfected? Or they're more likely to be reinfected?
Yeah, so that's a great question as well. There's been some studies looking at, infected oysters are more susceptible, or environmental conditions can make oysters more susceptible to infection. Different environmental conditions like, increased pollution or things of that nature. And so the treatments, while they get rid of the worm, they don't get rid of the blister. And there's also potential for reinfection. So another kind of future research area could be determining kind of the spawning season for worms in the area. If we know that, then we can maybe have a better timing of treatment application. If we know that worms usually settle around this time period, then that's probably the best time to also apply those treatments.
So that means that infestation happens in the larval stage of the worm?
Right, during settlement, yeah.
Okay, and then the blister, once it's developed, is a permanent feature, even if the worm is removed?
It is, yeah. So the oyster kind of continues to grow shell around it, and some of the older blisters are less visible, but they're still there, yeah.
Okay, we are getting a couple of other questions. So another one from Liz, are you aware of any misconception or knowledge from the public? What is the awareness from the of this outside the oyster industry?
Yeah, so we've been really cautious about, so there's not really an outcry from the public right now. We've been working on some of that public, consumer outreach materials, but some of the project collaborators are wary of releasing some of these materials before there's even an outcry. We don't want to, create more of an issue than it is right now. So yeah, that there currently isn't an outcry. But it's good to be cautious and take precautions in that way. So
aware. So we've got a question here is, and this is again looking on both sides of us here in Oregon, but is there a difference in infestation rate between farms in coastal bays and estuaries versus those in Puget Sound? And it was a little bit talking about some of those variability between temperature, salinity, those kind of things. Was there a pattern that you could pull from our other sample sites outside of Oregon?
That's a great question. So we haven't, I don't remember the exact distribution for Washington off the top of my head. But like I said earlier, it's definitely pretty variable, even seasonally, between each of across all of the farms. And part of the larger project, PIs in Washington are going to be comparing satellite data for temperature conditions and with infection or infestation. So we haven't fully teased apart those aspects in environmental condition aspects yet.
And he added
the
eel crust or eel toast is delicious.
It looked very delicious, yeah.
We have a question from Steve. You've been careful to refer to the polychaetes as shell boring polychaetes. Can you share what we know about possible species in this group?
Yeah, so there are several species of Polydora that are shell-boring polychaetes. The most notorious one is Polydora websteri. It's the most commonly researched and found worldwide. But the project team through Species ID has identified several Polydora species. I can't remember all of them off the top of my head. Within Oregon, Polydora websteri though is the most common.
So I had one more question for you, and you kind of alluded to it in talking about susceptibility of infestation maybe changing with environmental conditions. But I was wondering if there's any research around the age of the oyster itself and whether young oysters or older oysters are more susceptible to that infestation.
Yeah, that's a great question. I haven't personally seen research related to that. And our kind of initial hypothesis was that the larger oysters would be more infested because we were thinking of it as an age relationship. The longer that they're in the estuary, yeah, but we're seeing that they're actually smaller, and so maybe it kind of goes along with that. Mudworms might be causing decreased growth, but yeah, that's a great question as well.
So no laboratory work done yet, all field observations at this time.
Yeah, at least for the West Coast, yes.
Seems like there's a lot more work that needs to be done to answer a lot of these questions, and this work has been really foundational.
Yeah,
yeah.
Definitely a
So
baseline study.
I'm not seeing any more questions come in, but I am seeing thank yous and virtual claps for you. So thank you so much for taking the time to share with us. Any last thoughts that you want to share with your Oregon group before you head off and do other things?
No, thank you. Thanks so much for coming to the presentation. This has been great.
Perfect. All right, still getting thank yous in the chat, which I think you can see. So for everybody online, thank you so much for joining us at our last seminar for a little while. And again, keep an eye out for announcements for when we're going to start back up again and what format those will be. For Megan, again, thank you so much for taking time out of your busy schedule to be with us and sharing this really great work. Much appreciated.
Thank you.
All right, everybody. Until next time, thanks.